So this video is going to talk about the amino acid metabolism. So a little bit of review. Amino acids are the building blocks of proteins, and we have about 20 different amino acids in the body. And about 10 of those amino acids can be made in the liver, and we call them non-essential amino acids. The other 10 are called essential amino acids because we can't make them, and so then we must obtain them from the diet. Now all 20 of these are going to have some common or basic elements. They're going to have a central carbon, they're going to have a carboxyl group, and they're going to have an amine group. And all 20 of them will have those same things. An amine just means that it's going to contain nitrogen. But then there's going to be about 20 different R groups, and that is kind of like the rest of the molecule, and that's what shows the variation. That's what makes it each a unique amino acid. So here we have our central carbon, and there's a hydrogen on here. This is the carboxyl group. Here's the amine group. There's the nitrogen right there. And then this R group, that's the one that shows all that variation, gives us our 20 different amino acids. So still reviewing here, talking about the primary, secondary, tertiary, and quaternary structure of proteins. The primary structure of a protein is just the order of amino acids in the chain, in the protein chain, and that is determined by the DNA sequence. And on your DNA you have a gene, and genes are all of the nucleotides needed to make one protein. So when your cells decide that you need a protein, it's going to send a message to the DNA, it's going to copy just that gene onto messenger RNA, it's going to send it to the ribosome, and then that ribosome is going to make the protein, and it's going to put together the amino acids in the primary structure. Then we're going to form the secondary structure, and there's going to be some hydrogen bonds that form between the different amino acids, and that can either form an alpha helix, the one that looks like a little ribbon here, or a pleated sheet over on this side. And the pleated sheet kind of reminds me of in the summer when you're hot and you fold a piece of paper up to make a fan. It's kind of what this pleated sheet is going to look like. Then we might also have a tertiary structure, and this is going to be the final 3D shape of our protein. And in this example here, we see there's an alpha helix, here's a little alpha helix, here's another one, there's a longer one kind of behind this heme unit, there's another one coming up and down this way. And so this 3D structure is the result of interactions between the protein chain and any surrounding water molecules. And many proteins end right here with the tertiary structure. But some proteins continue on to form a quaternary structure because that one protein chain can't function all by itself. So the quaternary structure is when two or more protein chains interact with each other. And so this is an example of hemoglobin, which is called a globular protein because it's kind of in a glob or more circular. And it had two of those blue amino acid chains, protein chains, and then we also have two kind of purple here. Now really, in real life, in your body, it's not going to be color-coded like this, but this gives us an idea of what these chains are. But all four have to be present and functioning to make a functioning hemoglobin. We could also have an example here of collagen, which is a fibrous protein. It's all very long, like a yarn or string. All of it are some alpha helixes, but you have to have all three of these protein chains to have functioning collagen. Now, the more complex your protein structure, they can be destroyed. So denaturation is the destruction of the tertiary or quaternary structure of a protein. And that destruction is permanent. Things that can cause that are high temperatures or acidic environment. And for our discussion, the acidic environment is going to be very important. So from the time you eat something that has proteins in it, or amino acids, this is where we're going to talk about how we are going to absorb, break down, and then use these amino acids. So when you eat something, you ingest it in the mouth, and with your mouth you start chewing, and your saliva is going to mix with that food, and you're going to form a bolus. But that chewing is going to help break up some of the tertiary and quaternary structures. Then after you swallow, that food gets into the stomach. And there's some mechanical processing continues. Obviously you don't have teeth in your stomach, but we do have an extra layer of muscle. We've got this inner oblique layer here, and so with all three of these layers of muscle contracting, we have some more mechanical processing going on. The stomach acid is going to denature some of those proteins, that tertiary and quaternary structure. And then we actually have our first enzymes that are going to start breaking down proteins. Pepsin is going to start attacking specific peptide bonds. Where this comes from is pepsinogen. And pepsinogen was an inactive form of the enzyme that was produced by the chief cells in the gastric pits. And then that pepsinogen is going to be converted to pepsin by the acidic environment, by the hydrochloric acid in the stomach. Once the food has been mixed with the gastric juices, now we call it chyme, and that chyme is going to leave through the pyloric sphincter and it's going to enter the small intestine, specifically the duodenum here, this first portion. And when food enters the duodenum, the hormone CCK, or cholecystokinin, is going to be released, and that's going to stimulate the pancreas to release all of its pancreatic enzymes, again in an inactive form. And once they are secreted into the duodenum, they are going to be activated there. That begins with an enzyme called enteropeptidase. That's going to activate trypsin. And then trypsin will in turn activate a bunch of other enzymes. For example, chymotrypsin, carboxypeptidase, and elastase. Now each of those enzymes will attack a specific peptide bond, and the peptide bond is the what we call the bond between amino acids. And so as you attack those, what's left are tripeptides, or 3 amino acids, dipeptides, or 2 amino acids, and some individual amino acids. As this chyme then leaves the stomach, leaves the duodenum, goes into the rest of the small intestine in the jejunum and ileum. Most of the absorption of nutrients occurs in the jejunum. And the final step here is going to be taking place by these brush border enzymes. So here we have a cell of our jejunum with microvilli on the top, and so there's going to be some enzymes here located on the microvilli, dipeptidase, that are going to break down the remaining dipeptides. They're going to break them down into individual amino acids. They are going to be absorbed through diffusion into the cell, these amino acids. Then they are going to diffuse out through the rest of the cell. And then these amino acids are going to collect in the blood vessels and then they're all going to combine to form the hepatic portal vein. And that hepatic portal vein will deliver all these amino acids to the liver. Once they get to the liver can use these amino acids to make some plasma proteins. One of them, for example, is albumin. You may have heard of that before. But the liver can also help synthesize, convert, or catabolize amino acids. So when we're going to synthesize an amino acid, and remember there are those non-essential amino acids, the first process that we're going to talk about is amination. And that just means adding an amine group to an acid. So here we have a keto acid, or an organic acid. We add an ammonium ion to it, and now we have formed glutamic acid, or an amino acid. Another way to make an amino acid is through transamination. And here we have that glutamic acid, and another organic acid, could be a keto acid, and we use an enzyme called transaminase, and we just moved those amine group and a hydrogen over to that organic acid 1, and now we formed tyrosine, which is an amino acid, and we are left with a different just organic acid. Now if we can go through amination or transamination, that is making some of these non-essential amino acids. Remember that essential amino acids you cannot make in the liver, and so we have to get them from the diet. If your body is going to use amino acids for energy, we can catabolize an amino acid. And that begins with a process called deamination. So again, we're going to take that glutamic acid and we are going to remove the amine group. But instead of adding it to another organic acid, we're just going to take it off. And so that's left with that organic acid and the ammonium ion. And this organic acid, sometimes we call a carbon skeleton. And so let's talk about what happens to this carbon skeleton first. Now this is an example. Here's pyruvate, acetyl-CoA, and then this is the citric acid cycle. So you should have talked about this already. But we don't really need to worry about whether these amino acids are glycogenic, ketogenic, or both. But what we want to see is that some of these amino acids, when they undergo deamination, can then be converted to pyruvate and then enter the citric acid cycle. This list here can go directly to acetyl-CoA and then go into the citric acid cycle. These four are all converted to glutamate, which then enter the citric acid cycle down here, and so on. So they can be used in several different places in the citric acid cycle, which then provide the hydrogen ions for the electron transport system, which then makes our ATP. However, the body does not like to use proteins or amino acids to make energy because remember that we had the carbon skeleton, but we also made the ammonium ion. And ammonium ions are toxic to the body, so we need to get rid of them immediately. So in transamination, the ammonium ion is removed, but then added immediately to another organic acid. In this case, when we are catabolizing or breaking down amino acids, the ammonium ion must be converted to urea. And so we do that through what's called the urea cycle, and this occurs in the liver. Now urea is then excreted in the urine, which kind of gives it its yellow color, and it's harmless when it's in the form of urea. So I hope this helps you understand the metabolism of amino acids. Most of the images and text come from the Martini-Obernath book called Visual Anatomy and Physiology, and the amino acid catabolism image came from Wikimedia. Is a commons image.